Hi everyone, my name is YeeAnn Chen. I currently work in Dr. Lin He's Cancer Research Lab, and today I'll be going over a project I've been working on for the past year, and that's characterizing the tumor-suppressive function of miR-449 microRNAs in the Emu-myc B-cell lymphoma mouse model. So first off, many of you may not know what microRNAs are. In fact, I didn't either until I joined this lab. But what you may be familiar with is that that DNA contains genetic code, which is then transcribed into RNA and then eventually translated to protein, a functional unit. MicroRNAs are small non-coding RNAs, typically around 20 nucleotides long. MicroRNAs are processed from RNA and have many different functions, some of which actually inhibit this translation process. MicroRNAs are shown to be dysregulated in cancer, which is the premise for my project. Before discussing about miR-449, I would like to bring up another set of microRNAs, miR-34, that led us to study miR-449. miR-34 was one of the microRNAs that was discovered in a screen for microRNAs regulated by p53, a tumor suppressor protein. p53 is very important because it's actually mutated in about 33% of all cancers. There's many different stresses that can activate the p53 pathway, one of which is oncogenic stress. Oncogene meaning cancer gene. The oncogene of interest that we're studying is called Myc. This leads me to talking about the model that we use in our lab called the Emu-myc mouse model of lymphomagenesis. We chose to use this model because it has been well studied for p53 and its effector targets. What Emu-myc means is that the Myc oncogene is placed under the immunoglobulin heavy chain enhancer of B cells. This fused transgene causes Myc to be expressed in B cells. So mice that have this Emu-myc transgene will inevitably give rise to lymphoma, a type of blood cancer in the lymph nodes, within 6 months. So Myc helps cells proliferate and also induces apoptosis. Proliferation means the dividing of cells, and apoptosis means cell death. These two things depend on the different conditions, one condition of which is p53. If the p53 pathway is disabled such that it doesn't undergo apoptosis anymore, and yet the cells keep proliferating, this can lead to cancer. And I wanted to reiterate this point because I'll be going over it again throughout the presentation. So if cells are proliferating but not undergoing apoptosis, you can imagine that basically that the cells are growing and dividing, increasing in number without any sort of regulation of dying when they should. And so this leads to increased growth and tumors and eventually lead to cancer. So bringing this back to miR-34, miR-34 is thought to be a tumor suppressor because it actually inhibits the cell proliferation process and induces apoptosis, preventing cancer. So we set out to see, well, if we knocked out or deleted miR-34, would we see an acceleration in lymphomagenesis? So miR-34 has 3 microRNAs, A, B, and C. miR-34A is transcribed from chromosome 4, and miR-34B and C are transcribed together from chromosome 9. We're ultimately interested in seeing what would happen if we knocked out all 3 of these microRNAs, but on our way to getting there, we first generated miR-34A knockout mice, and then miR-34BC knockout mice to see what would happen first. When we knocked out each of these microRNAs, we saw that the mice did indeed die faster. And this can be shown through a survival curve. In the survival curve, the x-axis indicates the days survived of the mice, and the y-axis indicates the percent survival. Each vertical line down here indicates a death in a mouse. The black line refers to Emu-myc wild-type mice, and the red line here indicates Emu-myc mice that have 34A knocked out. So as you can see, the miR-34A knockout mice tended to die faster, and this indicates an acceleration of lymphomagenesis. We did the same for miR34BC, and we also saw this acceleration as well. So, in summary of what I've been saying so far, basically when you knocked out miR34A, we saw an acceleration of lymphomagenesis. So we knocked out miR34BC, we also saw the same. So the idea was that if we knocked out both miR34A and 34BC, that we would see an even greater acceleration of lymphomagenesis. But what was super surprising was that when we deleted the entire miR34 family, we did not see this acceleration. It was a very what moment. So, as you can see from this survival curve, there does not seem to be a significant difference between wild type and complete miR34 knockout mice. So ultimately, our project turned into asking, well, why does knocking out all 3 of these microRNAs from miR34 eliminate this acceleration of lymphomagenesis? Here's where miR449 comes in. miR449 is another set of microRNAs that belong in the same family as miR34. So we thought that perhaps miR449 was actually compensating for this complete loss of miR34. We hypothesized the pathway to be such that miR34 inhibits MYC, which induces E2F, a set of transcription factors, to transcribe 449. So if we knocked out just one of miR34, maybe that only slightly increased MYC and E2F. But when we knocked out both, perhaps MYC and E2F are greatly increased, upregulating expression of 449. So we first set out to see if MYC actually does upregulate 449. The way I studied this was that I did a qPCR on MEF cells, MEF meaning mouse embryonic fibroblasts, a type of cell line that we often use to study knockout mice. And I saw that when we induced MYC in MEF cells, we saw a 3-fold increase of 449 expression as compared to wild type. This suggests that MYC does indeed indirectly upregulate 449. What correlates with MYC upregulating 449 is that if you notice over here, the Emu-myc premalignant B cells, premalignant indicating before cancer formation, is that there seems to be a higher expression of 449 as compared to wild-type B cells that don't overexpress MYC. And as you can see over here for the Emu-myc tumors, the expression of 449 seems to be lower than wild-type B cells. So this suggests tumor-suppressive functions because it seems necessary to lose 449 in order to form tumors. Although we don't have a complete knockout of 449 mice generated yet, we do have mice of 1 copy of 449 knocked out, and we termed this 449 het. So this is a survival curve, and we saw that 449 het mice did indeed die faster as well. So we wanted to see why, and so we did tumor tissue stains. So over on the left here I've compared wild type and 449 het tumors. Up on the top is an H&E stain, and what's important to just see from this is that there seems to be a lot more holes in the wild type than compared to 449 het. These holes being dying cells. There's actually a term for it called starry sky because it looks like a starry sky. And I also quantified this, the cells dying with this immunohistochemical stain for caspase-3, an apoptotic marker. Brown cells here indicate positively stained for caspase-3. I've quantified this in the graph shown over there, showing that there seems to be a significant decrease in caspase-3 positively stained cells in 449 het as compared to wild type. Overall, this indicates that deleting 1 copy of miR449 results in a decrease in apoptosis. And if you recall from earlier in my presentation, decrease in apoptosis is one of the factors that can lead to cancer, answering or explaining why we saw an acceleration of lymphomagenesis. So I also looked at proliferation, and this time staining for phospho-H3, a proliferation marker. I saw that there was an increase in phospho-H3 stained cells in 449 het as compared to wild type. So ultimately, in answering our question, we want to see if 449 is indeed upregulated when we knock out miR34 completely. So I did a qPCR on mast cells once again, and I saw that when miR34 is completely knocked out, there seemed to be a 2 to 4-fold increase of 449 expression levels as compared to wild type. So this is very exciting and promising towards our hypothesis that 449 does indeed compensate for complete miR34 loss. So in future directions, what we hope to do is to generate complete miR449 knockout mice where both copies are knocked out, and then see that once we do so, does the reverse occur? Does miR34 get upregulated? Another thing that we're interested in is if we completely knock out everything altogether, miR34 and miR449, would we finally see this even greater acceleration of lymphomagenesis that originally hypothesized to happen just for miR34? So that concludes our presentation, and I want to give thanks to everyone in SURF, to Thye Fund for monetary support, for Nathan and Leah for running the SURF program, giving me advice. Also everyone in the Hull lab, Dr. Lynn Ho is my professor, my awesome grad student Margot Bennett, who's been with me every step of the way, and also the rest of the Hull lab, and especially to Nico, Byron, and especially Priscilla for giving me so much advice on everything. And also thank you to everyone who came out today.